Right, we ready to go?
Yes.
We might as well. It's raining. I don't think anybody else is going to show up. Yeah. A nice slip of paper because I need it. Right. Thanks for coming. Welcome to Life Inside and Out, which is our new biology department speaker series. Thanks to Bradley for getting it going. My pleasure to introduce you guys to Tamika Land. She's come over here from, she's from Tasmania. She's doing her postdoc right now at the University of Arkansas. The data that she's going to be presenting to you today is from Australia, from Griffith University. This is where she did her PhD. This is actually your PhD work. All right, take it away.
Thanks. So Carl already did a great job of introducing me, but in case anyone is geographically challenged, that's where Australia is. And the island down the bottom is Tasmania, so where I'm from. So I just wanted to give it, a little brief introduction about my research, my background. So in Tassie, I started off working on platypus, which are aquatic mammals. Moved up to Queensland to start my PhD on flying foxes, which are quite large bats. For my PhD, I also got to spend some time at the University of Cambridge. And then, as Carl said, now I'm at the University of Arkansas, go Razorbacks, doing my postdoc. And then for that, I spend some time in Kenya studying Bombali Ebola virus in little bats. So today, I'm going to be talking about some of the work that I did with my PhD and talking about, kind of the broader project that I was involved with and who I still work with. And the reason that I wanted to talk about that is that it's a really neat story about One Health, which is this kind of idea that human health, environmental health, and animal health are all interlinked. And that's kind of a bit of a buzzword. It does get kind of overused a little bit, but this story that I'm about to tell you about Hendra virus in flying foxes is actually a genuinely really good example of One Health. So to introduce you to the bats, because I know that you guys don't have these kinds of bats here, flying foxes are a type of fruit bat. So they belong to a particular genus, Pteropus. They eat pollen and fruit, and they are really important pollinators and seed dispersers. And these guys roost together in trees in big aggregations called a roost. And these can range from, hundreds of bats into historically millions of bats all roosting together in this one area. And if you've ever seen bats on the internet really enjoying watermelon or bananas, it's these guys. So in Australia, we have 4 species, the black, grey-headed, little red, and spectacled flying fox, and they have kind of various distributions across Australia. And then as this talk is about, these guys are reservoir hosts for Hendra virus, which causes lethal disease in horses and humans. So the fatality rate is, I think, 80% in horses and 60% in humans. So it gets passed from the bats into horses through contact with infectious urine, and then transmitted from horses to humans through contact with bodily fluids. If anyone is familiar with Hendra virus at all, I just want to flag that I'm mostly talking about the prototypic variant of Hendra virus. A couple of 2 years ago, I think, we discovered a new variant, and so we're still trying to, get to know that variant. So I'm mostly presenting on this first one. So something that we know about Hendra virus is that it's really seasonal. So bats are shedding virus or excreting virus in their urine, mostly in wintertime, and this coincides with spillover, so the transmission of the virus from bats to horses and then potentially onto humans. And so spillover coincides with kind of pulses of shedding. So this graph kind of illustrates that. So on the y-axis, we have the probability that urine samples will test positive from bat roost. And then on the x-axis is just time, and then the blue bars is winter time. So you can see that there are distinct pulses of shedding in winter time. So this knowledge has really helped with kind of telling people about risks of Hendra virus and how to mitigate those risks. So there's a vaccine. So around winter time, there's kind of renewed articles and news articles to say it's time to vaccinate your horses. But unfortunately in Australia, the uptake of this vaccine to horses has been quite low for various reasons. And so that's not a 100% kind of mitigation for this disease. And so we really need to know kind of mechanisms to be able to better predict times and places where people might be at risk or horses might be at risk. But so far, we don't know really the mechanisms that drive these pulses. So we know that they happen in wintertime, but they don't happen every single year and they don't happen from every single roost. So basically what the research that I've been involved with is kind of looking at this story of ecological change from bats and how this has impacted Hendra virus. So historically, these bats would migrate large distances across the landscape, chasing like flowering, pulses of flowering. But Australia has a really bad history of deforestation. So in this figure, in the red, you can see deforestation fronts, in the green are intact forest. So you can see that Australia has quite a lot of red patches there. If we zoom in and compare it with the range for the flying foxes, you can see that it's really, this deforestation front is really overlapping with the areas that flying foxes occur. And really key to the story is that this has disproportionately affected plants that flower or fruit in wintertime. So these kind of plants are ones that grow on fertile land valued for agricultural and urban development. So 80% of winter flowering trees have actually been cleared from the flying fox's range. Something quirky about Australia is that trees don't always flower regularly every year. They might flower 1 year and then not for the next 3 years, and then it'll flower again. So 13 of the 52 diet plants for flying foxes in winter flower reliably every year, but 13 of 52 plants flower in winter, but only 4 reliably flower every winter, so every year. So all of this combined means that there's very little food for flying foxes available in wintertime. So this is kind of culminated in ecological change in bats. So as this food has become a lot more scarce, it kind of increases the energetic costs of flying foxes foraging, so making that big nomadic movement across the land. So bats are foregoing that migration and forming kind of resident small populations near consistently available urban food sources. So these are trees that people plant in their backyards, like flowering trees or things like mangoes, because we have those in Australia. And just to show some data linked to this, the plot on the left is a plot from 1998, and then on the right is from 2019. What I want you to look at are these little circles. So each little, I think I'm the right angle, each little circle is a flying fox roost, and then the buffer around it is their kind of foraging range. And so you can see in 1998, there are some flying fox roosts around, but not too many. And then in 2019, you can see, this explosion in the number of flying fox roosts. And this has correlated pretty strongly with spillover of Hendra virus into horses. So on this plot, the y-axis is the cumulative number of bat roosts, and then x-axis is over time. So you can see that each of those jumps in the number of bat roosts kind of links with this breakdown of these big migratory nomadic bat roosts down into these fission small populations. And then if we look at Hendra virus spillover events in the squares, you can see that these kind of are overlapping with these fissioning events. And this kind of story is being written up into a big mega paper. So if you're interested in reading more about that, keep an eye out. But digging deeper into some of the mechanisms driving this change in link with spillover, what we hypothesize and what we're looking into is that, if food is scarce in winter, the bats are moving into urban areas. They're now eating plants that might be with less nutritional quality. So non-native plants tend to have lower energy, high levels of plant secondary metabolites, and lower digestibility. So that might cause nutritional stress in bats, coupled with there not being that much food around. So that could increase the amount that bats are shedding virus. And this is something that we're looking at with kind of dietary analysis linked with cortisol for stress, and then also measuring virus with the urine. There's also a team kind of pairing this with immune responses. So immune responses are energetically costly. If you have an energy restriction, that might cause kind of a suppression of your immunity, so an increased infection risk. And so we have a team kind of looking at this with experimental infections in captive colonies. Another way that all of this ecological change could increase spillover is just, pure contact, increasing contact with horses. So if there are fruit trees in horse paddocks that provide food for bats, bats might come in, roost in the trees, shed virus, and then horses foraging below might be exposed and become infected. So we're also doing studies to kind of map the location and density of horse properties and overlay that with where flying foxes are foraging. And then we are doing flying fox movement studies. So putting GPS trackers on the bats and seeing where they're foraging. But those mechanisms aren't a full explanation of spillover events. So an interesting thing that we see is kind of repeat spillovers from particular roosts. So you can see this in this plot here. This is spillover events are in the stars, and then flying fox roosts are again in the yellow with their foraging area in those kinds of buffer zones. And so this is data from May 2017 to September 2020, which is our kind of main study area. And you can see that particular roosts are associated with multiple spillover events. So this is unlikely to be foraging because we actually see a higher prevalence of virus from these roosts, so compared to other roosts around. And it's not necessarily linked to food shortage because the neighbouring roosts would presumably experience similar conditions. So it could be that there are local features of the roosts. So this is kind of a main theme of what my PhD was about, was looking at kind of whether the structure of bat roosts might be having a role in infection dynamics within the roosts and their likelihood to lead to spillover. And this also feeds back into this idea of habitat fragmentation and changing bat ecology. So in kind of the intact forest, in the, historically when bats had a lot of space, they had a lot of trees to choose from, they could kind of spread out across wider areas. And so within trees, the number of bats and the density could be lower. But then when they're coming into these urban areas where there are less trees available, they might be confined to smaller areas and confined to, a very few amount of trees. So the density of bats within trees might be higher. So I was able to, build mathematical models to kind of show this theoretically using empirical data from bat roosts, real bat roosts. So I mapped roosts. So in this plot, you can see a part of a roost. Each of those grey dots are trees, individual trees. And so using like this model approach, I could kind of test different scenarios. So in this scenario of very few trees available, kind of putting in scenarios of low total abundance through to high total abundance. So, about 200 bats through to like 4,000 bats, and kind of spread them individually into the trees. And then kind of mix between scenarios where, bats are mostly mixing within their trees and not really moving outside of that, through to scenarios where they're, moving between trees a lot more. And comparing this to kind of a base model where we assume that bats are just completely mixing throughout the roost, which is a bit unlikely because it means that bats are more likely or as likely to interact with their neighbour on a branch as they are to a bat on the complete other side of the roost. And so then I was able to compare, this sparse kind of structure with only 4 trees to an intermediate kind of structure through to a dense structure, which would reflect kind of the historical tree availability for flying foxes. And then you can introduce one infected bat into the population and then firstly see, whether or not an outbreak happens. If that one infected bat can pass on the virus to anyone else. And then you can also, if the outbreak is successful, you can have a look through the simulations to see what the epidemic is like. So you can look at the magnitude of the peak, so the proportion of animals that became infected, the timing of that peak, the duration of that peak, how long was the epidemic, and then the time to extinction if the virus became, worked out of the roost. So I won't go into this too much, but basically what this plot is showing you is the dark blue is if an outbreak failed, and this bar is showing you the dense kind of tree structure. So you can see that, and this is the proportion of simulations. So you can see that for the dense tree structure at this, like 2,888 bats in the population, you can see that most of the outbreaks did not, that there was no outbreak. If you look at an intermediate tree structure, there was also a high rate of just failure in the outbreak. If you look at the sparse tree structure, all of a sudden there's a lot more outbreaks successful, and then in the heterogeneous example, that's even more again. And then If we look at, so that was for assuming that bats are mostly mixing within their individual tree. If we're a bit more flexible about that, so bats can move between trees, you can see that outbreaks become a lot more, outbreak success is a lot more likely, but you still see this pattern of the sparse structures having a much higher level of outbreak success. And then, yeah, that maintains. And then you can also look at features like the magnitude. So what I want you to look at is that for this blue bar, which is for the sparse tree structure, it had a higher level of magnitude. And then again, if you scroll through the how much the bats can mix in the population, the relationship is the same. And then for timing and duration, we get like a mix of patterns depending on how much you assume that the bats are moving. So it's a bit of a complicated slide, but things I want you to take away are that for the sparse tree structure, we had a lot higher outbreak success, and the magnitude of that outbreak, so how many bats were infected, was a lot higher, and the timing and the duration, if we assume, you know, an intermediate level of mixing, it was quite a fast epidemic, and the duration was quite short. So kind of what this means is that this might be a missing piece to this puzzle of perhaps local roost structure is a feature driving spillover likelihood, and in particular, spillovers might be higher for roosts with sparse tree structures because epidemics are more likely to occur and we get a higher proportion of bats being infected. Okay, so just to wrap that up into one slide, what we are seeing is this kind of change in ecology of the bats, breaking down from nomadic large roosts into permanently occupied small roosts. And this is linked with increased transmission within flying fox populations. So we think increased viral shedding and increased bat-to-bat contact, and then also increased transmission from flying foxes to horses and humans through increased bat-to-horse contact, which lead to spillover. Then I mentioned the story about One Health. So just to make clear about that, you can see that environmental health, so the deforestation, the fragmentation of the landscape, is feeding into bat animal health, which is then feeding into human health. And then just because I love this cartoon and I think that it's a really poignant way to summarize this, I'm just going to read this out to you. It says, the World Health Organization states that the sudden emergence of Nipah and other bat-related viral infections can be attributed to the loss of our natural habitats. As forests shrink, searching for food becomes difficult and stressful, weakening our immune systems. This makes it easier for viruses to grow, multiply, and spread using us as carriers. Isn't it a bit unfair to blame me for turning our health situation upside down? And thank you.
Thanks, Tamika. We're going to hold questions until after Scott's done, and then they'll just answer them all at once. So, big welcome to Scott Carver, professor at the University of Tasmania now. Congratulations.
Thanks.
Scott did his PhD at Perth, Western Australia.
University of Western Australia.
And then did a couple postdocs here in the US at Montana Tech, where the two of us started working together. And then also at the University of Colorado State. You've been in Tasmania 10 years now, and Scott's going to be talking to us today about mange and wombats.
Thanks, Kyle. Well, thanks everybody for having me here today. It's really great to come along and chat with you all. I've met some of you over the years a number of times. I often like to begin these talks if I'm in a disease session that has lots of bats by saying these are my favourite kind of bat. It's called a wombat. And this here is a female mother wombat with its large joey. They're called joeys and wombats as well, and they're very cute and fluffy. But wombats really get smashed around by this disease called sarcoptic mange. It's also called scabies in humans. You may have heard it called scabies. It's caused by the same parasitic mite, a mite called Sarcoptes scabiei. I'll talk with you a little bit about that. But before I begin, I want to say thank you because as a professor, I do some of the work myself, but also there's lots of other people involved. So there's lots of students who have played an enormous amount of role in the research that I'm going to present today. There's also lots of collaborators and colleagues that have played a role, and funding organisations from sort of government level through to smaller organisations. We work really closely with community organisations as well. So it's not just working with sort of funders, but actually working with wildlife rehabilitators across Australia. And I also want to acknowledge some of the Aboriginal people on whose lands we work. And in Tasmania, that's the Mulanina and the Palawa people, and the Widi Widi people in New South Wales. There's a stronger push in Australia these days to try and acknowledge the Aboriginal custodians of the lands on who we work and who have been dispossessed from those. So I guess I want to start by saying there's lots of threats to wildlife around the world. There's lots of factors that influence those. And one of those is infectious diseases. And often those infectious diseases that are really impactful on wildlife are invasive forms. And you can look at things like this is squirrel pox virus in the UK that's introduced to the UK by North American squirrels that were introduced there. There's amphibian chytridiomycosis, which has been introduced all around the world through a whole range of mechanisms. This is a chamois in Italy, in the Italian Alps, suffering sarcoptic mange, and bat white nose syndrome here, which of course many people will be familiar with. It's really hard to manage wildlife in diseases or diseases in wildlife, individuals and populations. So we're really, relative to humans and domestic animals, really not very good at it. And there's many reasons for that, but I often like to think about what are some of the features of wildlife that make this particularly challenging. One of them is that wildlife are free-ranging, so they're not sort of confined to an area, which makes it challenging to get after them. Often you can't detect all of the individuals in a population. That may be relevant if you're interested in managing a pathogen in wildlife populations, and consequently you can't necessarily manage the disease in all of the individuals that you're interested in. And this is my favorite one, that wildlife are generally really uncooperative. So this is me chasing after a wombat with my net here and trying to save its life, and obviously it's not game. So these are some factors that make pathogen management in wildlife a real challenge. And the pathogen that I've spent a lot of my time working on over the last number of years is sarcoptic mange, which I've already mentioned is caused by parasitic mite Sarcoptes scabii. We published a paper earlier this year showing that it was documented in nearly 150 species of mammal, making it one of the most post-generalist of mammalian parasites that we know of, maybe only outstripped by something like Toxoplasma. And that number's over 150 now already, so since we published this work. It's not necessarily endemic in all of these species, but it isn't a good number. One of the reasons why this parasite is so successful is that it seems to be able to persist through multiple modes of transmissions. This means it's just transmitted in different ways. So it's in some species, it's transmitted by direct contact among individuals, such as in your sort of more goat taxa. In some species, it's entirely either environmentally transmitted, so mites are deposited into the environment that other hosts come into contact with, which is what we think is going on with wombats. And some it's a bit of both, so like den living canines, for example. And in Australia, this parasitic mite was introduced from Europe to Australia by European settlers and their domestic animals. We've got records of convicts coming to Australia that had scabies. We knew that people were introducing dogs. They were really important for hunting during early colonial times. And they also had mange at that point in time. And since then, it has been introduced to a whole range of Australian mammal species. Koala, this is an agile wallaby, a swamp wallaby, a dingo. This is a southern brown bandicoot, a ringtail possum, ear-nosed wombat, also called a common wombat, and a southern hairy-nosed wombat. And it presents this interesting epidemiological situation across the country, where in northern Australia, it's really a disease of humans and domestic dogs, and that's quite typical for the tropics for scabies. And in southern Australia, it's really a wildlife health issue, particularly in wombats, in koalas, and increasingly in quenda, which are southern brown bandicoots. And today I'm going to talk to you about ear-nosed wombats. Okay, so 3 species of wombats in Australia. There's the bare nose, the southern hairy nose, and the northern hairy nose. So the bare nose have the southeastern distribution, southern hairy nose more arid adapted, and the northern hairy nose are critically endangered and really just from one population here in Queensland and recently been translated to another. They're all fossorial mammals, so they dig burrows and live in those and come out and forage at night time. They're herbivores. So the distribution of Venus wombats is southeastern Australia, and we know that sarcoptic mange occurs right throughout that distribution. It's relatively well understood and has done for probably the better part of a century. And we think that the transmission of this parasite is via environmental means because wombats are relatively solitary animals. And they sleep down these burrows, and it's often just a single wombat in a burrow. But there's lots of burrows around, and they will switch burrows every 4 to 10 days. And another wombat can come in and use that. So we often call that sort of asynchronous burrow sharing. And mites can fall off wombats in these burrows, such as down in these bedding chambers. And laboratory trials suggest they can last up to about 19 days off the host. So if you've got wombats switching every 4 to 10 days, and you've got sort of a period of time where the mites can be potentially transferred via the environment to another individual coming in. But wombats it's a really significant welfare issue, this disease in wombats, and occasionally a conservation issue. So wombats go from being healthy to showing very clear signs of alopecia, so that's loss of hair and thickening up and scaling of the skin, and that leads to cracks in the skin. It's really quite an awful disease, and they ultimately die. These pictures were drawn for me by some elementary school kids in Tasmania, so I always try and show them in my talks. And it has really severe individual effects. We've done lots of studies on that, but I guess a couple of ones that I like to often point to is that you can score mange in these wombats observationally by their visible sort of how much hair they've lost and how skinny they're getting. And we call this a mange severity score. So 0 is healthy, going higher and the score is more severely manged. And in a healthy wombat, we spend 2 to 4 hours a day foraging, so out eating grass, and then we'll spend better part of 20 hours a day sleeping down a burrow. It's a pretty good life. But a mangy wombat will spend up to about 14 hours a day foraging. So it's a huge behavioural change. And one of the reasons for that is they're losing an enormous amount of heat to the environment. So it's through hair loss. This is a thermal image of a wombat. So the redder areas are heat loss, whereas it should all be more or less blue because the fur, protects that heat loss from happening. And their metabolic rates are also increasing as their disease severity increases as well. And that means they also need to forage more to meet the metabolic demands of this disease, which they generally fail at. So we were quite interested early on to sort of try and understand what's going on with their populations. This disease causes such significant welfare issues. Is there sort of a larger issue? And so this is Tasmania. All these different sort of coloured blobs are different meteorological regions across the state. And all these little nematode-looking lines are 10-kilometre driving transects that the state government does every year and counts all the wildlife they see, including wombats. So we basically interrogated that information. They've been doing this since the mid-80s for 130-odd transects. It's really a valuable data set. And actually, wombat numbers since the mid-80s have been trending up in terms of observations on these, and we've sort of calibrated this in lots of different ways. So we're pretty confident that this is at least a good index of population size. And it varies when you break it up into these different areas. You get sort of different patterns that you see, but overall it's this general increase, except for maybe the central northern population, where we actually have witnessed a mange outbreak and a local conservation issue. And this is actually what got me to start working on this. It was at this national park where we had an undergraduate field trip that was going on in northern Tasmania, just point to it, called Norontapu National Park. It was formerly farmland that was handed back to the Park Service in the 70s, and it's just a great haven for marsupials. So there's lots of kangaroos and wallabies and paddy melons out on these fields at night time grazing on grass. And in Australia, we call that a marsupial lawn because they'll mow it to like this high, and incredibly good lawn mowers. And wombats were formerly very abundant there. And we had a field trip from one of our undergraduate classes that used to go out there and count the number of kangaroos and wombats each year. And what we were seeing is that from east to west across these grassy areas, we were seeing wombats declining sequentially. So they're declining earlier in the east and declining later in the west, but they're all declining. And because they have these visible signs of alopecia, or hair loss, we're able to basically map this and actually look at the change in the distribution of that. So you can see these sort of orangey areas are basically the ones that are showing signs of mange, and the yellow ones are the healthy ones. And you can see that orange is moving from east to west across a part of the park there where we were able to monitor that. And we're able to show that as mange prevalence increased in one of these areas, each of these areas, the number of wombats would go down. So we're really able to clearly connect that this disease was what was driving this population decline. Now, we've got the situation where it's really widespread. It's not causing population-wide declines of wombats, but you can get these local conservation issues where it basically kills everybody off. So what's going on? How are we getting such weird sort of epidemiology? We've done a bunch of different modeling on this. This is just some outputs from one of the models that I want to present, where we're trying to look at sort of stable states. And mathematicians like these things called stable states, and these are mathematically stable states, not necessarily what ecologists might. But one of those is that we found is that you can get cases where all wombats and all mites die, so everybody dies, that's stable. Another one, which is our ideal conservation scenario, all mites die and wombats survive. Another one where you get coexistence between the two below sort of a theoretical carrying capacity of wombats in the environment. And then you can get this other one, which we call unstable coexistence. So it means they're fluctuating like this. And empirically, we see 3 of these in nature. So we're starting to get good evidence now that this actually sort of works really well. And the determinants of that seem to be the number of wombats in an area, but not just the above ground abundance of wombats, but it's that ratio of wombats to burrows, because that influences the encounter rate of the mice in the environment. And we're still getting our head around this one a bit more. We know that wombats switch burrows every 4 to 10 days. Things we don't know so well, or didn't know so well, was how long mites can survive down those burrows, and how many wombats are actually basically dropping into the environment. So we had a go at this one recently, which has been really fun. This is one of my honours students' work. She went out and started mapping wombat burrows. This is using ground penetrating radar, and you can see in the picture on the right-hand side, a tape measure just basically mapping out this burrow from above ground. And then we worked with a robotics engineer at another university in Australia to develop this robot that could traverse wombat burrows underground, which is really quite tricky environment to get down. We call this the Wombot. And then we sort of took this laboratory data on the relationship between temperature and humidity on how long mites survive, which basically at low temperatures and high humidities might survive longer in the environment of the host, fitted a statistical model to that, and then came up with some estimates at different times of the year. So what Liz found was that we estimate that mites can survive up to about 16 days during the winter down these burrows, and as low as about sort of 5 or 6 days during the summer. So we think there's really strong sort of seasonality in the transmission happening via the environment there, which is probably quite important for the dynamics that we see in nature. So we're working on that. We take videos, because we can on this Wombot, and the university stitched that together into a little 1-minute video. So I thought you guys might enjoy this. So that's that video went really well. The university put it online, and my last count had like 5.2 million hits. It's like the most popular video the university's ever produced. quite entertaining. But it's a really fascinating world in these burrows. It's really much more complex, and it's really hard to design something that can actually traverse the really difficult terrain or complex terrain underneath there. So I guess one of my main questions I'm often interested in is, can we manage this disease in various forms? And our first crack at this was at Ngururruntupu National Park, where we had a treatment that we knew worked for curing wombats of mange disease, but it has some limitations. So we had this drug called Cydectin, or the chemical name is called moxidectin. People put it on their cows and stuff. It's really common agricultural ectoparasite and parasiticide, and also for worms as well. And we know in wombats from some pharmacokinetic trials that last about 5 days, so you have to give them a treatment of this, but the mites can survive for 6 to 16 days down these burrows, and wombats keep moving among them quite frequently. So you have to basically retreat wombats, which is tricky. And they have lots of burrows, and these were just all the burrows in the area that we were working with in the national park there. There was over 200 burrows there. In reality, there was over 600, but we thought that 200 of them were active. So basically what we were trying to do was trying to treat every single wombat in this area every week for 12 weeks, healthy, mangy, the whole lot, and try and see if we could create population scale, disease resistance, and see these mites die out in the environment, maybe even lead to eradication. And we were doing that with these, what we call burrow flats, which is basically super simple. It's a wire frame that goes over a wombat burrow. It's an ice cream container lid that hangs down from that, a little milk bottle cap that's slotted into that. And as the wombat walks out, Cydectin is poured into that, and it pours on them as a little pour-on dose. It's quite ingenious in its simplicity. And here's myself and my PhD student at the time looking in an area where there's lots of wombat burrows. This is a Lynn actually installing one of these burrow flaps. We worked with a wildlife rehabilitation group called Mange Management. So lots of people from the community actually go out and treat wombats for mange in Australia, and people feel quite strongly about that. The drug company gave us all the Cydectin. Here's a wombat walking out underneath the burrow flap from one of our Canberra traps. We did sort of simple things to try and sort of like tying up the zip ties really tight. So once the burrow flap got pushed up, it stayed up, so we could determine how many of them, which burrows were active every week. Sometimes this worked really well. So there's a wombat going down into a burrow, getting a dose of Cydectin. Sometimes not so well. There's wombats not very happy about our burrow flap. Sometimes they would even rip them out of the ground. So it created some challenges, but we were seeing it work in the population. These are a couple of individuals, so they're kind of mangy on this end. Then they look worse in these next photos, but that's actually after all the scabs, all the infected areas fallen away from them. This is healthy skin underneath, and eventually their fur regrows. And this is some of our data. So the blue shaded area is this 12 weeks of continuous treatment in this population. The black line is the number of wombats we surveyed each week. The red is the number of infecteds, and the blue is the number of ones that were showing signs of recovery. So you could see we were having an effect. We were exerting a population scale disease management, but it's only with a huge amount of effort. It's an enormous amount of effort for a Lynn undertaking this disease control work. It's not sustainable. And ultimately, when we stopped, what we saw was the disease come back. We weren't able to eradicate it entirely, and the population continued to decline. Now, a lot of people sort of finish off there when they undertake disease management in wildlife and go, well, we tried, it didn't work. We gave it our best shot. I think we can do a bit better. So we've been trying to basically learn from that. So we've been trying to sort of create new models that work better for our system. We've been using a state-based model framework, which we fit to our data from the field. And when we use that to ask questions, what are we missing? What can we do better? So one of the things we learned early on was that we actually think we're only delivering the treatment about a third of the time successfully. It might be that the treatment actually hits the fur but never makes it to the skin to absorb into the animal. It might be that you get some wombat that goes and sticks its head down lots of different burrows and gets heaps of treatment, and then lots of other ones miss out. And we saw both of these things in the field. And they're both probably relevant. We can ask questions about how we can get better at this. So on this plot here, these 3 different lines just represent different model fits to our data. The x-axis is the improving probability of delivering treatment successfully, and the y-axis is prevalence of disease among the burrows. So what we're seeing is that, and the black dots is about where we think we are. So if we get better at delivering the treatment, we think we can have a better chance of controlling it. If we have a longer lasting treatment, remember our treatment only lasts 5 days, which is pretty short, then we also think we can get better at it. And even better is if we can combine these things together, we can have what we call a multiplicative effect. So a longer lasting treatment that we can deliver more effectively by the colors in these bars, better chance of reducing the prevalence. And so that's what we've been doing. We've been going forward into using a new drug called Bravecto. You can get it from the vets here for your cat or your dog as a spot-on treatment. We've been doing trials, pharmacokinetic safety and efficacy trials, and wombats on that. I've never thought I'd have to learn some pharmacology, but sure enough. So this is one of the curves of the drug being metabolized within the blood of individual wombats. This is time post-treatment, and this is the amount of fluralana or Bravecto that we're detecting in their blood. So you can see it declines, peaks, and then it slowly declines away. And to cut through all of this, basically, we think that a single treatment can be effective for greater than 30 days in a wombat. So that's 6 times longer than the previous treatment. So that's a pretty decent improvement. And we've now been going out to multiple field sites in Tasmania and in New South Wales to basically undertake sort of field trials. These are quite different sites with different epidemiology. These are some of my students myself. This is an anaesthetized wombat, one of my PhD students and former honours student working on this individual there. And this is a wombat that I treated in April, and then this photo is from June. And you can see this black outline. This is all the area where it's lost fur that was infected, and that's where the fur is now growing back. So single treatment, that's just a single treatment of this. So it can really help individuals recover quite significantly from this. And we're still processing the data from this at the moment. But I guess what I want to say is I want to hark back to some of those early points I made that you can't necessarily see every individual when you're working with wildlife. So for a subset of our wombats, we've caught, anaesthetized, tagged, and then tried to remonitor. So we only really see about 60% or less than 60% of our wombats that we've tagged again. And that's the same for control and treated individuals. And only about 45% of them we ever see more than once again. So there's quite a lot of work to basically get an idea of how effective your treatment is in the field. And so we've sort of asked questions using some models from this. So say if you wanted to be able to follow disease in 10 individual wombats, how many would you have to tag and how often would you have to undertake surveys to be able to do that? So we think if you wanted to monitor 10 individuals and you tag 20 wombats to begin with, you'd have to undertake sort of 10 to 20 surveys before you could at least be sure of reciting those 10 individuals more than once. And of course, those numbers change based on whether you tag more or monitor more. But our treatment's working. We're seeing as wombats that have a lower initial mange severity are more likely to fall into this recovered group, and ones that have a higher initial mange severity tend to be recovering, and they probably need follow-up treatments, so monthly treatments for 2 or 3 months. And I guess more importantly, we're really interested in what we can translate from this research. It's not just about the learning, but actually what can this implement into the real world. And one of the things that we've had this year has been the Australian Pesticides Veterinary Management Authority has granted the drug company what's called a minor use permit, which means that people from the community who are rehabilitators who actually go out and try and manage this disease in wombats can use this drug without veterinary oversight based on our research, which is a significant translation. We've produced our own sort of user-friendly guidelines because, of course, lots of people can't really read scientific papers and it's not reasonable to expect them to understand all the technical jargon. So we really try and sort of break through that and deal with like common questions that you get, which are very different from what a scientist would ask. So it's really important to sort of understand those differences. We've also been working with wildlife agencies that work with rehabilitators. So the drug company is sponsoring drug for free to these wildlife agencies that then they distribute out to wildlife rehabilitators through their networks. So it's really important translational work. And it's all with an effort of taking sick wombats like this and trying to return them to healthy individuals like this. I want to thank all of you. Any questions?
I'm curious if over time you've seen selection for wombats that change their growth less often, if there's like 2 different groups in the population, if there's been selection done also for resistance to scabies at all?
So we're not yet seeing clear signs of resistance in wombats to mange. I'm not convinced that it doesn't occur. I'm not convinced that it does occur. What we really need to do to answer that question is to go into endemic populations where it's endemic and try and do antibody studies. We just haven't managed to do those yet. At the moment, we sort of work on this assumption that once they get it, they progress to say disease and death. And that's generally supported by evidence, but there could be resistance. What was the first part of your question?
Has there been any change like over time since this was introduced fairly long ago?
Yeah.
in whether or not they switch burrows a lot, like maybe a long time ago, they switched burrows more often and there's selection for morphs that don't switch burrows?
That's a good question, and I don't think we have any evidence for that either way. Yeah, it's a great question.
Is there a difference in the incidence of this mange at different elevations? I noticed that I think you look like you're almost coastal, and you're up there in the very northern part of Tasmania, where it's, remind me what the name of the place is where they have the Tasmanian devils that, you know, you can watch them at night. What's that called?
Up at Cradle Mountain area,
Yeah.
things like that. Yeah.
Well, Cradle Mountain, area, when we were there, just before COVID hit, they all look like this. I didn't see anything with
Yeah.
mangy at all. And-
Yeah.
And they're a high elevation, it's cold up there like it is in Montana. What, is there less mange in high elevation?
Generally, yes. So I've got a student at the moment who's trying to pull apart all of that at sort of landscape scale. And I mean, the general pattern that we're sort of seeing is lower elevation, more likely to be agricultural areas. We see more mange. Is that because it's just colder, or is it because other factors? I'm not entirely sure, but that's generally the pattern that we're seeing. Yeah.
I guess, is there like any kind of like other long-term goals you plan to get out of this research that could apply to other species, especially species of concern that have the problem with mange?
Yeah, so certainly other species, other hosts of mange disease, this is really relevant for. And there's lots of people in different areas of the world that actually try and manage this disease. There's like a reasonable amount of work in black bears that's occurring at the moment. And, you know, there's some people who are treating black bears with the same treatment that we're using. There's people doing trials with ibex and chamois in Italy and in Spain. There's people interested in foxes and kit foxes in Bakersfield. There's a population there in California that we're having like a real bad, hard deal of it. It's recently disappeared in that population, which is, I don't entirely understand why. Might be aridity or something like that, but they've had some significant problems there, and they're trying to manage it as well. So there's lots of actually cases. So so we're trying with what we do that it can apply to other ones as well, and potentially in other systems. I should say that Tamika will take questions too. If anybody has any back questions.
I had a back question.
Yeah.
I remember you mentioned that there's different sizes of the bat roost populations. There's like some that were 100, some like 1 million. And then you did the mathematical simulation where you had sort of different sizes. And I was wondering, was that all homogeneous sizes that you looked at, or was there variability?
Yeah, so I just chose particular scenarios. So I choose a scenario. My lowest was like 400 bats in the whole population, and then I think I went up to a maximum of like 5,000 or something like that. Because the roosts that we see nowadays don't get anything close to what they used to be. So they used to be, I think, in the millions, but now it's real. You definitely get higher than 50,000, but yeah. So it was those select numbers, and then that distributed amongst the trees in the model. Yeah.
And was it the same numbers, or was there like a variation in size?
It was the same numbers, and the same numbers per group of tree. Yeah.
And in reality, is there a fairly large spread in the sizes of the different groups, or does it tend to be clustered?
So for the kind of roosts that are in intact forest, they can spread out across much larger space because there's a lot more forest available for them. For the ones that are kind of roosting in urban parks, they're a lot more restricted. So for the models, for the data is just like a 20 by 20 meter plot. So the spatial kind of spread that the bats are in the models are fixed. But in reality, yeah, it could be spread over a much larger space.
Do you think it would be interesting to model like more complex network structures in there, where you might have, like dense populations that disperse a lot, that are like super spreaders versus ones that tend to be isolated and the different levels would be connected to one another? Yeah.
So, I mean, we kind of got it a little bit, but, you could definitely, I think with models, you could do, anything you want with them basically. And it's just a matter of, deciding what you want to look at basically. So we kept it fairly simple and picked, scenarios that reflected the empirical data that we had. But you could definitely make it more complicated and explore it that way.
You answered her question. Any other questions?
I guess both.
You mentioned that, I think the transmission was, you said something about trees with fruiting trees like mangoes or something, above where there are livestock like horses.
Yeah.
Now, the people, are they catching this from eating the mangoes that are getting urinated on, or are they getting it through the horses?
For this particular virus, Hendra virus, it's just through the horses.
And how do you get it from horse?
You just mostly vets get it, and it's through like interacting with them too closely. So bodily fluids from the
Fluids
horses.
not having gloved completely or
Yes.
something?
Yes. So since Hendra virus was discovered, there's been, major kind of change in veterinary procedures around horses for mainland Australia where there are flying foxes, because, they used to go with not that much PPE, but now if it's a suspected Hendra case, they are wearing full kind of hazmat suits to be handling them. Yeah. And I mentioned that there's a vaccine, but the uptake of the vaccine is pretty low. Vets also sometimes will completely refuse to see a horse that hasn't been vaccinated because the risk of them, contracting the virus is high, and it's not a good virus to get. It causes like respiratory and neurological, like very severe disease.
Mm.
Yeah. Oh, but another thing I wanted to say. So there's a related virus called Nipah virus, and it's spread in a similar way. So contact with infected urine, but and this occurs in like Bangladesh and Malaysia. And in that virus, humans can get it directly from the virus, from the bat. So there's
From the urine of the bats?
yeah, from the
Not
urine of
biting?
bats. So no, not biting. It's mostly through contact with urine. So bats will kind of feed at date palm pots. So people, are harvesting the syrup from date palm trees, and they have pots to do that. And the bats will also like it because it's sap, and so they will come in and urinate in the pots, and then people will drink it as well, and they don't treat it because it's just a culture of this is how they've always done it, so they can drink it and get it directly from the bats that way.
All right, this is a question for the both of you. Do you highly recommend like studying abroad in wildlife and disease management, like just getting out or getting a hold of other universities for like a seasonal application?
Yeah, I've had a really great time being able to travel with my research, and have those experiences. Some of it is voluntary. I guess I've travelled less abroad for sort of voluntary related research, and more like within the institution for those aspects. And the travel components have come through different stages. So I did a degree and masters in New Zealand, and I went to Australia to do further studies, and then came to the US and did further studies, and ended up back in Tasmania. So that's how it's happened for me. But just the general experiences in each location, people are always looking for volunteers or things like that. It's been really great.
Yeah, I would say the same thing. I've gotten to move around a lot. For me, even moving from Tassie to Queensland was a big deal for me in the beginning, and then getting to go to England, that was really cool. And now I'm doing field work in Kenya and like living in the US. So moving around definitely gives you a lot of exposure to meeting lots of different people and hearing different ideas and perspectives about like problems and animal kind of problems. And the experience that you get with kind of seeing and handling different animals and like working with different pathogens and things, I think it's just broaden your understanding of issues and what you're interested in.
Yeah, I was wondering, somewhere early in your slideshow, you showed bat roosts and then outbreaks. Were those outbreaks measured in the bats in the bat roost, or is that when the horse got sick?
That's when a horse got sick or a human got sick.
Right.
Yeah.
And so, it seems reasonable that it comes from the closest bat roost, but also bats and flies. Do you have any sense for like how good of an assumption that is?
Yeah, so there is data to support that it comes from the closest roost. So the way that bats kind of forage is that if they're staying in a particular roost, they will forage in an area and then go back to the closest roost. And so you would not see a bat going and foraging somewhere, bypassing a bunch of roosts get to the roost that they wanted to be in. Like they are foraging where their roost.
So those little like Harmony diagrams you draw.
Yeah, exactly.
Accurate to life.
Yeah, so when we see a spillover case, we kind of assume, we do assume that it's linked to the closest bat. And we have done, as part of the study, we've done kind of like spillover response sampling. So if we are notified that there's been a horse case, we'll sample a bunch of roosts in the vicinity and just like see what the prevalence is. But
But you do occasionally get a bat that moves from one roost to another and
Yes,
spreads it
yeah.
across the roost.
yeah, and but bats are, some bats are resident, and so they will stay within the colony and then they won't move. And then some bats are nomadic and they will leave. So it really depends on the bat.
So for the bats, when the disease is transferred through the urine, how long is the disease present around their roost after kind of a infection around?
Yeah, so the length of time that the virus can stay kind of viable in the urine is only a couple of days. But the amount of time that the virus might stick around a roost really depends, because it depends on, which bats are getting infected. And so, we really don't have a good idea of it. It could be a matter of weeks, it could be longer than that. And that's kind of what we were trying to get at with the spillover response sampling. We would, come back repeatedly, but there were only, I think five spillover events during this period of study, so it's kind of hard to statistically say a lot about 5 spillover events, but it's probably in the realm of like a couple of weeks to a month that the virus might be sticking around at a roost.
And why is it mostly high in winters? I missed that. Why does it go up in the winters and then decrease to almost nothing in the summer?
So we think it's mostly because of the food availability. It's quite low in winter time.
Density of
And
bats.
so it makes it denser
Yeah, so
transmission.
it could be that there's not food available for them, and so their immune system decreases. So their immune response, decreases, and so they can't like fight infections very well. They might become more susceptible if their neighboring bat they get into contact with the viruses, so they might be more susceptible. There's also another really cool thing about bats, which I didn't mention at all, but there's a theory that they kind of host kind of latent infections over time. So they might have a virus, but sustain it at a really, really low level, so not actually shed it, and they just kind of tolerate it over time. And then the theory is that if they get stressed or something like that, something happens to them, their immune system stops working so well, and that might give the virus an opportunity to kind of infiltrate them a lot more. And so then you might see kind of that might be when you see it shedding from the urine. So the main hypothesis that we have is that it's to do with not much food availability that causing stress, but that's something that we're looking into.
And you said that introduced trees that flower in, cities and things are really attractive toward people and livestock. But is there any, I don't know if you would mention this, that there was any attempt to try to put flowering trees back out on the landscape and get them spreading back out, or is this irreversible now that once they're in the cities, you're done, that it's not gonna spread back out?
So the kind of next part of this research agenda is what we call like ecological intervention, and it's exactly what we're trying to do is plant trees back out of cities that we know that flying foxes love. So there's a team within the project who are working with people who are already planting trees. So there's a big system in Australia of replantation, particularly geared towards koalas. So replanting trees because everyone loves koalas, and so like boosting koala numbers. So we're working with these like kind of koala people to say, these trees are also good for flying foxes, good for koalas, and also good for flying foxes. So these are the species that you should be planting outside of urban areas. And so that is something that we're working on, but it's going to take time, for the trees to grow and then become mature and flowering. But
And it's eucalyptus then, that some of those are flowering.
yes.
Okay. I wondered what kind of trees flower. We have some kind of trees that have flowers,
Okay. I think
Eucalyptus
all eucalyptus
but
flower.
it's all eucalyptus. They do,
And they flower. So there are a bunch of different species of eucalyptus. I think there's
like 100.
like Yeah, there's a lot of species.
Unbelievable.
And they all have their own kind of flowering dynamics. So some of them flower in summer, some of them flower in spring, and then, yeah, as I said, some of them flower in winter time. And some of them are more regular flowering, some of them are pretty irregular, they're very picky about their conditions. And so, yeah, we're kind of targeting especially the eucalypts that flower in winter time and that flower regularly in winter time, so that, boost the food availability.
Maybe the last question, Neil? Oh, I was just wondering, does horse behavior change season at all? Like, you play, like, get left out of pasture and have more chance to, like, eat a fruit in the winter, or is that not really an issue?
Maybe. So I don't know. I'm not a horse person, so I'm not 100% sure. But some of the kind of risk communication to horse owners is to tell them, keep your horses away from fruiting trees so that they don't come into contact with horses. Put shelters over your feeding troughs and water troughs so that bats don't accidentally urinate into them. So horse owners know the things to do to kind of mitigate their horse risk, but whether they follow that and whether that varies across it might, but I'm not sure.
All right, thanks, guys. All right.